All right, let's get started. Welcome, everybody. It's great to see everybody here. And welcome to the event that we call Raising Up the Next Generation of Aging Research. It's actually our first event of its kind, so you're here at the inaugural event, and we really want to thank you all for joining us today and supporting us in this new endeavor that's entirely driven by our group of trainees that not only the biggest group of employees here at the Buck Institute, but also a really talented and very, very driven group. So we're really excited to share with you our program today. So my name is Malene Hansen. I'm the Chief Scientific Officer and also a professor here together with Gordon Lifko. We were actually really excited to be asked by the trainees to help put together or host the event. Both Gordon and I run research labs. We study the biology of aging, which you will hear a lot more about. But then we also share a passion for education and training and the upbringing of the next generation of researchers, which we're all here today for. So we were really honored to be asked to be involved with the event. We're truly blessed working with a really, really talented group of trainees, and as you can see here at the bottom, they're organized into groups of very, very, very prolific nature, and you will get to know a lot more about them. So there's a group for students, there's a group for postdocs, and we have a multicultural group again that you would be able to learn more about after this seminar session. So yeah, again, we appreciate your support and being here today, taking your time to learn more about the book. And so maybe I can start by asking by a show of hands, how many of you have never been to the book before? Always great to see new faces and learn of new friends and supporters who can spread the word and actually share our joy for science. So what is the Buck? The Buck started almost 20 years, a little more than 20 years ago, and was the first research institute in the United States that was dedicated only to understanding the biology of aging. And really the key, as you will hear from the talks, is that aging is the common denominator for many of these diseases that I'm sure you all know. So the premise here is that we study aging as a common process for all of these diseases, and with the mission to eventually learn enough about aging that we can also not only help combat one, but many of these diseases at the same time. That's sort of our mission in a nutshell. It's not a small vision at all, and it will take a lot of effort to get there. And it really does. So what we do is a lot of science. What we really measure to that end is like a lot of scientific papers. This is our product, if you wish. We do research, we write it up with our trainees, we share it with our collaborators and with the rest of the scientific field, and this is how we build on the science and hopefully get further and further in our understanding. Like I said, we have a lot of collaborators, and perhaps even more importantly, we have a really thriving community here at Buck. So there's more than 200 people working here. I would say the biggest group, again, is our junior scientists, our trainees. More than 80 people are in that group and are really the workhorses for everything we do here. And what I also think is a cool comment here is that we're really diverse. There are people here from over 30 different countries. Myself, Denmark, Gordon, actually Scotland, so that's 2 of them already. And so it's just a very, very exciting community to be part of. Of course, it costs money, and so we rely on money from incoming sources, including the government, which I'm sure you're aware of is an increasingly difficult task. But what do we do? So in this vibrant community, we do a lot of things. We do events like today. Many of those, we love having guests and supporters come visit us. And of course, the crux of it is the science that we do. So people in the lab that actually do the research. And so really excited to share with you the stars of our research in 3 talks today. So the program today is we'll go through these. They're kind of short, and then afterwards we'll have a poster session and reception where you will have a chance to talk to even more of the trainees. And many of them have box shirts on, and they have a little tag, so feel free to ask the scientist anything you kind of want. We're a basic research institute with translational aspiration. That means we do not do medical research per se. We do everything behind medical research up to clinical trials. We're happy to talk more about that. But the talks today, very exciting, and it's actually from 3 different types of precision, if you wish. So the first talk in our keynote will be from Bedoya Sen. She has a PhD, meaning she's already a trained scientist. She happens to work in my lab as what's called a postdoctoral researcher. She would like to run her own lab one day, and so she's training for that, kind of like a doctor in residency, if you wish. So she will give the keynote. I'll introduce her in a second. Ariel Floro is a PhD candidate, so she's training to get her PhD, to go on to do all the many cool things you can do afterwards, academic science, biotech, writer, venture capitalist. There's a number of career tracks to be open for Ariel once she's finished. She does work on immune aging in our CEO's lab, Eric Burden, he's here. And then last but not least, we'll hear from Max, who is a postdoctoral researcher. That means he's just before making the decision if you do want to go towards graduate school and become a PhD. And so it's a nice sort of range of position, if you wish, within our training community. And Max will tell you about space flight and how that relates to aging, so very cool. Without further ado, I'm just going to turn over the podium to Bejoya, but I want to preface it and just introduce her a little bit. She's a postdoc in my lab. She's been at the Buck for 5 years. And like I said, she has a PhD already, which she got from the Institute of Liver and Biliary Sciences in New Delhi. And she's going to tell you about the exciting science, no bias, of course, in our lab, that focus on how cells in our body are actually able to recycle and in that way kind of keep themselves useful and basically better for longer. So without further ado, Bejoya, the scene is yours.
Okay. Good morning, everyone. My name is Bejoya Sen. As Malena already introduced me, I am a postdoctoral researcher in the Hansen Lab, and today we are going to talk about brains cleanup crew and the importance of taking out the trash. So before we dive in, let me tell you who I am. So I was born and brought up in the eastern part of India, and I got my master's in biotechnology from Amity University. I then got my PhD in biomedical sciences from Institute of Liver and Biliary Sciences in New Delhi, and I studied how increase in the number of aged cells in the liver may lead to progression of liver cirrhosis and liver cancer. And then in 2019, September, I moved to the Buck Institute to do my postdoctoral training in aging biology. I came here with my husband. We love hiking. This is our picture in Yosemite. We now have almost 2-year-old. He runs around in the house, gives random hugs, and then would say, not I love you, but I love you. But the reason that I joined this aging field of research is my grandma. She is currently 86 years old and has now started forgetting things. Let me give you an example. In our video calls to India, so let's say, for example, I'm talking to her for about 10 minutes, she would ask me at least 2 to 3 times if I have eaten or not, because she forgot that she has already asked. And not only that, she sometimes forgets that she has a grandson. So, as you know, that aging is the major risk factor of various chronic diseases such as diabetes, osteoporosis, cancer, sarcopenia, and neurodegenerative diseases such as Alzheimer's and Parkinson's. And we believe in the book that studies on the biology of aging will provide insights to help combat these chronic diseases. Neurodegenerative diseases are characterized by cognitive decline with symptoms like poor judgment, asking repeated questions, having trouble thinking, misplacing items, and forgetting past events or names of places and objects. So before I tell you what we are doing to address this problem in our lab to help my grandma and others with the same condition, let me ask you a question. What would happen if we never took out the trash? Our home would be a mess, right? That's where household recycling comes in. And the same exact process happens in our body in every cell. And this process is called cellular recycling or autophagy. This process actually helps every cell in the body to recycle the trash so that it can function properly and normally. So let's break down this process. We have a plastic bottle, right? We use that plastic bottle, it's normal, it has a normal function. We crush that plastic bottle, make a pile of it. And this plastic bottle, the trash, is now identified by a hand which would pick this trash up, put them in the recycling bin. And the contents of these recycling bin are then taken up by a dump truck to a recycling factory where we get a fresh new plastic bottle. And this exact same thing that I just explained happens in every cell in our body, be it a brain cell, a heart cell, intestine, muscle cell, liver cell, skin cell, you name it. Every cell, this process happens, this autophagy process happens. And in fact, it is happening right now, especially if you are a little hungry. So what actually needs recycling? Anything damaged or anything in excess, such as excess cellular components, needs recycling. For example, this plastic bottle is now damaged and is in excess, so it needs recycling. And in the cells, autophagy actually literally mean self-eating. So auto means self and phagy means eating. So here a cell, when it is starved, it eats its own components such as cellular organelles or proteins to make energy to proper function. So what are actually proteins? Proteins are the building blocks of the body. Let's take an example of this paper, right? When you fold this paper into a proper structure, it can do its normal function. Whereas when it is not folded properly, it becomes a trash with no function. And that's when autophagy or the recycling process comes in to pick up this trash and make it into a better, normal functioning protein. So what do proteins do? Proteins are of various types. There are digestive enzymes, which are proteins, which helps digesting the food that we eat. There are antibodies, which helps with the immune function. Then there are hormones, which help regulate the body. There is hemoglobin, which is also a protein, which carries oxygen and transport things throughout the body. And there is proteins which are required for structural support, such as collagen. And this neurodegenerative diseases feature protein aggregation. For example, you can see here this normal brain with a healthy neuron. So neuron is a brain cell. And then with age or in patients which have these neurodegenerative diseases, there are evidence of aggregated proteins, which is a toxic buildup of these excess damaged proteins. And this ultimately leads to the disease and symptoms of a neurodegenerative diseases such as Alzheimer's. One of such proteins that gets aggregated is tau. So tau normally has a good function, such as it maintains the neuronal structure, It facilitates transport of nutrients and neurotransmitters. So neurotransmitters are molecules which are moved from one neuron to the another so that it can pass information throughout the line of the body so that we function, we do our job that we are doing. And with age, or in patients with neurodegenerative diseases, this protein, tau protein, forms an aggregate and accumulates in the brain. That's when autophagy, or the cellular recycling process, comes into play and degrades these aggregated proteins and makes normal functional tau. However, with aging, this process, the cellular recycling process, declines, and ultimately we get neurodegeneration. So let's see how this process actually happens in normal conditions. We take our favorite example, the plastic bottle. So let's say this plastic bottle is the tau protein, the normal functioning tau protein. With age, this tau protein forms aggregates, right? And these aggregates are now identified by a hand, such as anchor proteins. So it's now identifying these aggregated proteins and putting them in the recycling bin, which are autophagosomes inside the cells. And the content of these autophagosomes are taken up by a dump truck, which are motor proteins, to a recycling factory, which are called lysosomes. And these lysosomes have enzymes which can degrade these aggregated proteins to make normal functioning tau protein. So as you can guess by now that I study this process in context of neurodegenerative diseases. So how do we study autophagy experimentally? I can study this in my grandma who has this disease, or in my son who doesn't have this disease, right? So we need model organisms to study this process. And we have this small nematode, Caenorhabditis elegans. It is the simplest multicellular organism to study aging and age-related neurodegenerative diseases. It is very small. For example, this small petri dish can hold about few hundreds of these worms. And to give you an idea, here is the human hair compared to the worms that are moving in this petri dish. They live for a very short time, which is approximately 2 to 3 years. And more importantly, the cellular recycling genes that are required to pick up this trash are present in these worms. And we can engineer these worms to express human disease proteins such as tau. And when these proteins are expressed in these engineered worms, we get the appearance of aggregates within 5 days compared to humans, where it takes about 60 to 70 years to form these aggregates. And then, so we can study them faster and multiple times, right? Our Hansen lab had made an observation that in worms engineered to express those disease proteins, if we increase the number of the hands that can pick up the trash, this aggregation becomes smaller in number, which would mean that the big trash of plastic bottles is now smaller because it is getting put into the recycling bin faster. And not only that, these worms are longer lived and even healthier. So this is already done in our lab. What am I doing? I'm trying to find new types of hand that can pick up this trash. So instead of picking trash by one person in a beach, like this kid picking up plastic bottles in the beach alone, we would have multiple hands picking up the trash so that there is faster recycling of this trash. Okay, enough said. Why should you care, right? As I mentioned, these protein aggregates are a feature of neurodegenerative disease, which is a toxic buildup of this trash in the brain. And if I find, or when I find more hands that can identify this trash, we might be able to find drugs that can boost these hands so that it can pick up that trash faster, and ultimately we would have a normal neuron or a brain cell which is free of this trash, and then we would have alleviated the symptoms of neurodegenerative diseases. But we are not there yet, right? This is gonna take a lot of time. So I'm studying this process in C. elegans, or worms, which have roughly about 60 to 80% similarity to humans. I would have to do these experiments in mouse models, which have about 85% identity to humans. And then we would have to find drugs, which would be this drug discovery process. And I would have to test these drugs if they are safer in these model organisms. And ultimately, there would be human clinical trials in different phases, like phase 1, phase 2, phase 3, and then we would have a drug in hand that would finally help combat this neurodegenerative diseases. And we are only here. We haven't even started the drug discovery process yet. So as you can see, this is gonna take a lot of time, effort, and money. And this means that I cannot help my grandma right now, but I might be able to help my son in the future. Okay, so this is gonna take time. What do we do now? We can right now activate our cleanup crew. And two things that are known to activate this cellular recycling process are exercise and calorie restriction. These two processes mimic the kind of starvation that can happen in the cell. And when the cells are starved, they would eat up the damaged proteins, aggregates, or components that are in the cell to build up better components. So right now we can do these two things. With that in mind, I would like to thank our team in the Hansel Lab, which works on this process, autophagy. And I would like to thank everyone in the Buck Institute who works hard every day to combat these aging and age-associated diseases. And lastly, I would like to thank every one of you for coming here and your attention. And I'm happy to take questions now.
Thank you. Thank you so much, Victoria. We have time for maybe a few questions. I also want to mention we have a lot of people on the Zoom call. If you have a question, I would encourage you to write them in the chat and we'll answer them later. Any questions? No.
Yes.
So what sort of chemicals have you found that are in your studies that are actually working with autophagy?
So right now, we are trying to find those hands. So that's like anchor proteins. So they're not chemicals. We are trying to identify targets, which are proteins that can pick up these aggregated proteins. And ultimately, our goal would be to find chemicals that can boost the making of these anchor proteins in the cell.
But to answer your question, there are drugs that trigger autophagy. Yeah. So metformin is a known one, right, that many people take for diabetes. It's known to induce autophagy. Rapamycin is another safe drug to take. Does many other things. So it's not clear exactly what comes from what. We have personally been involved with collaborators to find some that are more specific that we're currently investigating in this sort of pipeline. Yeah.
To your knowledge, is there any approach in terms of trying to overexpress these anchor proteins in cells, in terms of just having a genetic modification of sorts in the cells?
Yeah. So the thing that I did not mention in today's presentation is we have neurons, like brain cells, in culture. So we can culture them in lab, which expresses those aggregated proteins. So we can target those proteins. So there are different kinds of model organisms or models such as cells. So your question is to see, like, instead of genetically engineering, if we can express those aggregated proteins in cells. Is that your question? Anchor protein. Oh, we can, yeah, in cells we can do it. So this would be like the data that I showed from the Hansen lab actually expressed those anchor proteins in that engineered disease model, and then it alleviated that, it decreased the number of those aggregated proteins. So, for example, let's say we all have a worm which has diseased tau, and if we engineer it again to express more of the anchor protein, the tau aggregate decreases. Yeah.
Maybe I could just add there too. Yes, there has been many examples, not many, but a few examples of overexpressing the same type of proteins in more complex organisms, and they have benefits there too. You can't measure lifespan in those, in some cell culture and also in some mouse models, but there seems to be benefits from that exact approach as well. So, good question. Okay.
So we have one Zoom question. Does calorie restriction mean maintaining healthy weight or being underweight?
Calorie restriction means maintaining healthy weight. So calorie restriction could be of different kinds. You could overall reduce the amount, like calories that you are taking, or you can restrict the time that you are eating, which would be like fasting. So some people do kind of like 12-hour fasting and eating only in the 8-hour of time. So that also triggers the body to go in the starvation mode, so that it can eat up all the things that are already there in the cell and make energy out of it.
All right. Let's thank Bijoya again and move on. Thank you so much.
That was fantastic, thank you. I take out our household garbage every morning, and as I'm walking down the stairs, I'm going, come on, brain, take out the trash, take out the trash. I'm delighted to introduce Ariel Floro from Eric Verdin's lab. Ariel graduated in microbiology, just like me, yes, immunology and molecular genetics at UCLA, and has been doing some phenomenal work in metabolism. And this is not new, but an increasingly important area of research, both here at the Buck and elsewhere, where the normal metabolic processes that we've been studying for decades are now seen to be really vital in the way in which we age. And when she's not doing martial arts and wrestling, she's doing fantastic work that she's going to tell us about right now.
Thank you. Okay. See if this works. Cool. All right. Hello, everybody. Thank you so much for coming in person or coming via Zoom. My name is Ariel Floro. I am a fourth-year PhD student in Eric Verdin's lab, and I'll be telling you all about my research today. And thank you, Gordon, for the introduction. I have some fun pictures to also demonstrate my introduction. I grew up loving science all throughout school. I actually was originally going to go to medical school because I was fascinated with the human body. But when I went to college at UCLA, I discovered research, and that became a much more interesting path for me at the time. So ever since college, I've been really interested in research. I've also been interested in the immune system, and those have been my passions in that way. And at the same time, I've also been very passionate about exercise. I wrestled in high school and college. I currently love skiing and rock climbing. And my interest in sports and activities have led me to an interest in nutrition so that I know how to properly fuel my body. And it felt like I had to keep these two passions side by side until I decided to apply for PhD programs. I decided to come to the Buck and study longevity research because we're classically told to eat well and exercise to live a long and healthy life. And I wanted to understand the biological mechanisms as to why that happens. I still study the immune system today. I find it very interesting. The immune system is a network of cells and tissues throughout our body that helps to keep us from getting sick. We could think of them as like little security guards that detect invaders and will get rid of them. So in a healthy immune system, we have these immune cells that will detect these pathogens, viruses or bacteria, for example, and it mounts an immune response to then get rid of these from our body, protecting us and keeping us safe from getting sick. As you would predict, with age, this immune system becomes dysfunctional. The immune cells are not as able to detect and attack these pathogens. That then leads older individuals more susceptible to becoming sick from these pathogens. And the way that we see immune aging manifest at a population level is two main ways. We see that older adults are much more susceptible to getting bacterial or viral infections, as I just mentioned. A big example of this is the COVID-19 pandemic, which was caused by the SARS-CoV-2 virus. Older individuals were hundreds of times more likely to be in the hospital or to die from the illness compared to younger individuals, thus demonstrating how important age is of a risk factor here. Unfortunately, at the same time, older adults are less responsive to vaccination. And you can imagine how this then exacerbates vulnerability to illness. If an older adult is already more susceptible to becoming infected with a virus, for example, they go in to get the vaccination to prevent them from getting the virus. But if they don't respond properly, then that just really leaves them more vulnerable to illness. So this is a really important and big, complicated problem that we're interested in. And the reason that we see these is because of changes in the immune system with age. There are plenty that are identified, but just to name a few, one example of an age-related immune change is chronic inflammation. And this is one that a lot of people may be familiar with or have just heard at some point in their life. Inflammation is meant as a temporary state. It comes and goes short term. It's meant to help our immune system to get rid of the viruses or bacteria infecting us. But in older adults, they may have this inflammation even at baseline. This is also a problem from when you are infected with an actual pathogen, you already have inflammation there. It makes it a lot harder to mount a proper response and clear out that infection. Another example of an age-related immune change is this increase in exhausted cells. And these cells don't carry out their typical functions as well as they do in their younger counterparts. So these are just two examples of the many age-related immune changes that happen. But you can see how this is a really important, complicated problem. Thus, we ask how we can alleviate aging in the immune system specifically. Unfortunately, we don't currently have any clear established interventions for immune aging specifically. For example, we know somebody with high cholesterol may go on to a statin to decrease their cholesterol levels, but we don't have that equivalent for aging in the immune system. So we're curious as to using certain interventions that might be promising in targeting the immune system, specifically in the aging that happens in this system. And when we talk about interventions for aging or age-related diseases, you may remember earlier that Bijoya had mentioned how aging is the #1 risk factor for chronic diseases or age-related diseases, some of which here, diabetes, cancer, and heart disease. So when we look at interventions to treat aging, we may look at their ability to treat one or more of these age-related diseases. And two really interesting interventions that are currently being researched to treat age-related diseases include fasting and ketogenic diets. I know these get a lot of buzz in the media, and I think they're really interesting. Fasting is just abstaining from food for a certain period of time, could be overnight to multiple days. A ketogenic diet is a diet that is meant to mimic fasting, so it's eating a low amount of carbohydrates and a high amount of fat to trick your body into thinking that it's fasting. But as I say these two dietary interventions, I bet you're thinking those sound awful and really difficult to stick to. Fasting just has to rely on your own discipline to not eat, even if you're hungry. Already doesn't sound fun. Ketogenic diet, while you can eat some food, you may have to cut bread and rice from your diet, which can be manageable. but some people may even have to watch their vegetable intake because even vegetables have some carbs in them, and then that already is decreasing the number of foods you can eat and is becoming really restrictive there. I think also with these two restrictive dietary interventions, it's unrealistic to ask older individuals to stick to things like this, especially as we know how important social life and social connections are for a long and healthy life, and think about how many social gatherings are surrounded around good food and drinks. And one of the ways that fasting and ketogenic diets work is that they tap into this evolutionary mechanism in our bodies, and they force our body to make these metabolites, which are called ketone bodies. But as we just went through, these are two interventions that are difficult to stick to, especially long term, or to have something be sustainable for a while. So our lab and the clinical research unit here at the Buck, including many other labs at other institutes, have been exploring the idea of supplementing ketone bodies directly. And so we've been looking at this in the form of a drink, which I'll refer to as a ketone body drink. And the idea here is to improve adherence and to see if we can have these drinks get the benefits of ketone bodies. They've been identified to have a whole host of benefits throughout your system. To see if we can have these alongside your normal typical diet instead of having to stick to these very restrictive dietary interventions here. So I mentioned our lab, including many other labs, are looking into the ability of these ketone bodies to improve aging and age-related diseases. And bringing in the immune system back into this, my question that I'm trying to answer during my PhD is if these ketone body drinks can be used as an intervention for immune aging. We completed our first clinical trial at the Buck, very exciting, about a year and a half ago. This was a pilot clinical trial where we were testing these ketone body drinks in older adults. And the main goal here was just to test if they were safe and tolerable in them. They are. That was a published article we had last year. So that's very exciting, and that then launches plenty more clinical trials that we can do here at the Buck, testing these ketone body drinks in humans right now. We also did some exploratory data on that clinical trial, and some of my preliminary data shows that the ketone body drink caused some changes in the immune system. So I'm following up on these results today, moving into a mouse model to see if these ketone body drinks can improve the age-related decline in vaccination response. So this is what I'm currently working on, but as I mentioned, we have this ketone body drink that we're doing clinical trials with now that has been launched off of this pilot clinical trial. We actually have 2 clinical trials looking at these ketone body drinks today. The STACK study on the left here is looking at the ketone body drink affecting different ages. Are there certain differences in people's lives that might affect the way they respond to it? And we have another one in older adults where we're looking at the ability of the ketone body drink to stave off frailty. So you scan the QR code here to learn more. We also have the clinical research unit at a table out there to answer any questions or sign you up for participating in the clinical trials. But this is really exciting work that we can do today, and it is a lot faster to get into humans. Bijoya had also mentioned how important basic science is, but those are about a 10 to 20 year timeline that we're looking at. The exciting thing about these ketone body drinks is that they are being done today in clinical trials, and once we get this research, we can clear up a lot of the questions that we have of how they can help treat aging or age-related diseases. So that's what I have for you today. Thank you so much for coming. Thank my lab, thank the podium pointers for helping me out, and happy to take any questions.
Questions, please. Yes.
I'm not looking for the recipe, but what do you put into these ketone drinks? I mean, what constitutes the ketones?
So ketone bodies themselves are actually very acidic, so you can't give those directly to people. So it's a precursor molecule that then triggers your body to make the ketone bodies.
Any other questions? Yes.
Are you using any probiotics that actually your body uses to create the ketone bodies to begin with?
That's actually an interesting point. It's still a bit unclear, but there is some emerging data showing that there's some bacteria in the gut microbiome that can promote certain ketone bodies or can actually produce them. So it's still very, like a lot of mouse work, and it's still really understanding it, but who knows what will come from that later on.
Anyone else? One more here. Thank you.
Did you find that some people that have that had COVID in 2020 or early COVID that got long COVID have that where like they're aging faster and having these age-related diseases?
Yeah, there's a lot of research, especially right after the pandemic, that was looking into that. But even having like mild or severe COVID, if you had one or more of these age-related diseases, then you were more likely to have a worse COVID outcome. And then especially with long COVID, people are kind of looking into what are the overlaps between aging there. So there is a lot of work being done into that.
Wonderful. Thank you very much. Great presentation. Okay, our next speaker is Max Manwaring-Muller. Max is trained as a molecular cellular developmental biologist at UC Santa Cruz. He previously studied breast cancer. He's a baccalaureate, as you've heard previously, in the Weiner and Furman labs. And Max has this trick. It sounds like magic. He can defy gravity. So if you find yourselves raising out of your seats, don't panic. It's just Max doing his great stuff. This science is literally based on things that happen out of this world. Max.
Thank you. All right. Hi, everybody. My name is Max Manwaring-Muller. As I was just introduced, I'm a post-baccalaureate research associate in the Furman and Weiner labs. And like Gordon said, I came from UC Santa Cruz, but I actually grew up in San Rafael, so very local. Grew up here, went to Terra Linda, swam and played water polo, moved down to Santa Cruz, and back here. It's hard to beat the Bay Area, so I'm really happy to be at the Buck. And to study such interesting things. I was drawn to the Furman and Weiner labs because not only were they studying really interesting aspects of immune aging, but they were thinking in really novel ways, trying to understand if we could come up with new models to study aging. And that has led down a really interesting path, as Gordon says, that's taken us out of this world. So we're studying astronauts as a model for accelerated aging. I'm going to try and convince you that that's a model worth pursuing, and I'm going to also show you some data that we have and an interesting study that was just published in Nature. But let's start talking about the current model organisms that we have. So like the past speakers have brought up, we use things like C. elegans or mice to study lots of aging disorders, anything from immune decline to neurodegenerative disorders. And that's great. We've learned so much from these models. But at the end of the day, if we want to study and really fully understand how to improve health outcomes for adults, we have to move and translate our findings into adults. So really, a lot of the amazing work that we have now to intervene for these age-related disorders has to get approved through human studies, and that's great, but it takes a lot of money, and it takes a lot of time, and it's really complicated to run these studies because humans exist and go and live in their normal lives, right? You can be part of a clinical trial, but then you go home, you're exposed to different pollutants, you eat different diets. I mean, there are so many factors involved in our lives that it's impossible to control for everything, which is why working with model organisms is great. But it also would be amazing if we could stick with the human system and just work on a more accelerated timeline. And that's where spaceflight comes in. So just really quickly, when you see an astronaut on the screen, that means I'm showing data from astronaut or studies in astronauts. All of this data has been taken from studies aboard the International Space Station. And these are roughly 6-month missions out to outer space. And if you see an older adult, well, we're talking about studies from older adults, both men and women over the ages of 65. So let's start with one of my favorite examples, a study in the musculoskeletal system. and how long we would expect it to take for certain declines to be observed. So when we're talking about skeletal muscle mass, the muscles that are responsible for moving our bodies, navigating, lifting our feet, right? To see a 10% decrease in this muscle mass, it would take, on average, 16.5 years. And there are definitely differences between men and women, but on average, in adults older than 65, that's how long it would take to naturally lose that amount of muscle. To see an 8% decrease in total bone density, something like osteoporosis, where our bones are more frail and less able to hold ourselves up, more likely to break in a fall, it would take around 20 years. That's a long time, and it's very hard to study these conditions if that's how long it's going to take to see these outcomes. Let's turn to astronauts. To see 20% decrease in skeletal muscle mass in 6 months is amazing. And to see, again, an 8% decrease in total bone density, osteoporosis, again in 6 months. And this is just the time duration of one trip aboard the ISS, one standard mission. And this is probably fairly intuitive, right? This is what happens when you unload your system. This is the byproduct of microgravity. But this story is not just true for our bones and muscles. It's true for a lot of other systems. And so I'm going to highlight a few here today. We're going to dive specifically into the immune system, but it's useful to talk about these others because it's really interesting how we're seeing a global accelerated aging effect in astronauts. So like I said, we see declines, notable declines in the musculoskeletal system. But when we talk about, let me go back. When we talk about the cardiovascular system, we see a drastic increase in arterial stiffness. We also see a drastic decrease in cardiovascular fitness. When we jump to the brain and our neurocognitive systems, we see increased, really stark increases in markers of neuron damage, things like tau that was just brought up earlier today. And we see an increase in brain swelling. And where our work focuses is the immune system. And so, really interestingly, a majority of us here have had some kind of infection in our lives. A lot of them, like if you get the influenza, right, your immune system is able to completely rid that of our systems. But there are some viruses that go dormant. They get beaten back by our immune systems, but they never fully leave our systems. One example would be Epstein-Barr virus, EBV. The majority of us probably have that living in our systems right now, and our immune system is doing a great job holding that back. But a telltale sign of immune decline is when these viruses reactivate. Your immune system is not doing a good job surveying. And interestingly, astronauts, around 95% of them have the EBV reactivation, meaning that their immune systems are suppressed in space. We also see a drastic increase in a lot of inflammatory markers, things that we see in older adults that tell us there's likely immune suppression going on. Now, why might this be? Why would there be an overlap between astronauts and older adults? It is not intuitive at first, but there's an overlap. And so when we talk about astronauts, there's only a few environmental factors that can be responsible for this decline. And we know that it's because of the environment, because these are perhaps some of the most healthy individuals. I mean, they have to go through such a rigorous screening process to become an astronaut. So they're physically and mentally very sharp and ready for a lot of stress, yet we still see age-associated declines. And there's only a few things, like I said, in space that could be responsible for this. So starting out, a sedentary lifestyle. What we see in space is microgravity. I mean, you're perpetually unloaded. You're not supporting the weight of your own skeletal system. And you're not having to put nearly as much effort into every movement. And so you see atrophy and you see a loss of bone density. But that's not it. And that certainly plays a pretty significant role, and we'll dive into that in a second on how that specifically affects the immune system. But there are very important other aspects, such as circadian alteration. Astronauts experience a sunrise and a sunset approximately every 45 minutes. Our systems are not evolutionarily designed to handle that kind of exposure and day, rotation. So, and then additionally, they're exposed to a lot of radiation in the form of galactic cosmic rays, which is very harmful and irritating and inflammatory. And they're also socially isolated. I mean, they're removed from their support networks, their families and friends, and put into a tin can in space. So that's very stressful. And we see that there are a lot of overlaps to those specific spaceflight environment factors in older adults. As we get older, it has been shown across many different countries and many different populations, we become more sedentary. Our circadian systems do decline. Our ability to go to bed and wake up at the same time every day, get good quality sleep, REM, declines. And we also know that not only does it decline with age, but you can accelerate that decline by perturbing that normal system, exposing yourself to blue light before you go to bed, not eating meals at the same time, or staying up late. Older adults accumulate a lot of exposure to radiation. I mean, your time on Earth, you're seeing the sun a lot, but also we see more than now than ever, we actually are screening and looking at to see how much radiation exists in our environment. And it turns out that 25% of Americans are exposed to a radiation, a gas called radon, above the accepted EPA threshold levels that are standard and healthy. And so we know that radiation is playing a role in our health trajectories. And also, as we get older, older adults are more likely and more prone to social isolation. And so that's why we think there's a significant overlap in these age-associated diseases. But this isn't just an idea. I mean, a lot of work went into proving and establishing that this could be possible. But we're not only thinking about that, but we're actually bringing it to our labs and using models to study specific facets of the spaceflight environment on Earth and see if we can use them to study how aging might be happening. And so one system that I really think is just ingenious, it's fairly simple, but it works really well, is the rotating wall vessel. And what we do here is essentially suspend cells of any type from any organ in media, which is just nutrient-rich water, liquid essentially, that keeps the cells happy. And we slowly spin it and put these cells in a perpetual state of free fall. Just like if you or I were to go skydiving and jump out of a plane, for that brief minute and a half that we're falling, we're experiencing microgravity. We're not loaded. Our system weighs basically nothing to itself. That's what these cells are experiencing. And so we can study at the molecular and cellular level what happens under the exposure or the environment of microgravity. And we also have amazing access to one of NASA's huge databases called GeneLab. And this has all of the data from many different space missions where we've taken blood or other kind of tissue from astronauts or some of the model organisms that were brought up earlier today, convert that sample into data that we can study on the computer and run analyses and statistical analyses on. And we can look for markers, biomarkers, or signatures of aging in this population, and we do see there are some. And so I'm going to tell you really briefly about one of those studies where we combined the rotating wall vessel with this bioinformatics analysis using statistics on the computer to understand the overlaps between older adults and astronauts. So to start, we took immune cells, because that's where our labs are interested in, is the immune system, and we spun them in the rotating wall vessel to simulate microgravity and see what the effects are. We then took every cell and looked cell by cell individually at the genetic activity. How was the cell responding to the environmental stress of microgravity? We're able to tell with some really, really cool technologies that we don't have the time to get into today. And we compared that signature, what was happening to the cells we spun, to data from the Inspiration 4, the JAXA, and the TWINS study, which are all studies of astronauts in spaceflight. And we saw that there was a really significant overlap between what we were seeing in our study in the rotating wall vessels and what was happening to the immune cells in astronauts. Specifically, what did we see? Well, we saw a lot of increased stressed protein production. We saw increased inflammatory molecules. And we saw structural changes. The immune cells are actually being shaped and shifted by the effect of microgravity. So great, there's an overlap. We validated that our model seems to do a good job of recapitulating what we see in space. Can we do anything about it? And do we think that this actually is going to lead to some meaningful interventions or discoveries? Well, Matthias, one of the postdocs in the Furman's lab, ran a drug discovery pipeline and found that one molecule in particular, quercetin, should be able to reverse a lot of the genetic activity that we saw in both astronauts and our rotating wall vessels. And interestingly, quercetin can be found in all kinds of natural and healthy foods. And so just to dive into the details a little bit more, we saw that quercetin reversed around 70% of the microgravity-driven genetic activity. We saw that it decreased oxidative stress, which is a kind of stress that accumulates in our cells and will damage the proteins, just like Bijoya was saying, when you have to recycle all of that protein, all those aggregates or trash in your cells, well, a lot of what's causing that damage is oxidative stress. And it also decreased markers of inflammation. So this, these are very healthy foods, and we think that this could play a role in helping dampen chronic inflammation. Beyond dietary intervention, though, what else is there? Well, to combat the sedentary lifestyle, it's super important to go on walks and do light resistance training. I think this is something that as we get older, we think we're supposed to avoid resistance training, avoid the gym. And, don't go start deadlifting like an animal, but it's really important to make sure that you're still moving and you're maintaining muscle mass and bone health. To keep your circadian system healthy, avoid blue light before bed and make sure that you're eating and sleeping, if you can, at roughly the same time each day. To avoid radiation, really check your radon levels and wear a sunscreen. And then to prevent social isolation, talk with friends, see family, and join social groups. It's super important to stay connected and to feel and to be healthy, to stay connected. So with that, I would like to say thank you so much for having me. It's been a blast to bring you all into the work we do, and thank you to my labs and the podium pointers.
Thanks, Max.
Oh, and I'll
got time for
I'll take questions.
Couple of questions. Yes.
Yeah.
Curious if I know protein folding is something that as we get older, we have a lot of that. Is that happening in space? I know mostly it's chemistry, with positive and negative and so on a protein, but does that actually happen in space to accelerate?
Mm-hmm. So let me make sure I'm getting your question right. So as we get older, we see an increase in protein misfolding, and do we see that same thing happening in spaceflight?
Yeah.
Well, I can't say that spaceflight itself increases protein misfolding. What I can tell you is that we do see an increase within the brain and in these cerebral spaces that hold cerebral spinal fluid, an increase in misfolded proteins. And so I don't think we're actually seeing an increase in the misfolding. I think we're seeing a decrease in autophagy and the clearance of these misfolded proteins.
Again, with the space flight, the reactivation of some of these viruses and so on, does this happen during the 6 months or always later?
Mm-hmm. Yeah, great question. So importantly, the model for accelerated aging would be in space flight, and we see the reactivation aboard the ISS. When they come back, interestingly, a lot of these astronauts return to normal health, which is a really good sign. It means that when you are in this kind of declined state, you can come back. And that's why it's really exciting to us, because we think that, one, if we can develop therapies to keep astronauts healthy when they're in the spaceflight environment, that's great and has a lot of translational potential to older adults. But it's also a message that if you go back to a healthy lifestyle, living as healthy as possible, you can see a huge recovery in things like the immune system and many other systems, yeah.
Question here.
Yes.
This might be silly, but how do you check radon levels?
Yeah, great question. Yeah, it's I don't know the exact specifics, although my dad is an architect and he says that they're supposed to check before you build new houses, but it is something that changes, and it's also, well, I believe, and there are studies, and you can find this on the EPA site. It's only checked in environmental hotspots or zones where they expect radon to be very high, but those maps actually are pretty outdated. And recent findings from Harvard have shown that it's much higher in places that we didn't expect radon to be there, which is why so many people are affected. I don't have the answer on how to check it, but I know that you can, and there are sensors out there. Yeah.
Two more questions. One here.
I was really curious that you said that astronauts within a short amount of time regain all of the normal functionality. So they are the perfect study models to look at how do their biomarkers or genetics or expression of proteins change? What has been seen in terms of what kind of modifications are happening there?
Yeah, that's actually a super interesting point. I'm more familiar with the changes happening in spaceflight, not the actual recovery process itself. But that's a great idea. I'll have to look into that more. I don't have an answer. That's a great idea, though.
And question here.
Yeah.
With the selection of astronaut process, is there reason to believe that there's biological superiority in that group?
That's an interesting question. I don't think so. I think that they are, they're definitely taking people who prioritize physical and mental fitness, are staying very active. However, from the selection process, I think they try and choose the best team because it's so important to have great teamwork out there. I don't think that they're definitely not running any kind of genetic screening or anything. It's really just who can survive stress the most. And I believe that can be really trained. I think most anybody could train for that level of fitness.
Let's thank Max and our other tremendous speakers. Wonderful. Thank you. Good job. Just a few brief words to close. First of all, there's actually a large number of people going to making an event like this happen. Our Graduate Student Society, our Postdoctoral Association, our Multicultural Trainee Association, the Philanthropy Department, and we'd all be sitting here in the dark without any slides if it wasn't for facilities in our IT department sitting up at the back there making it all happen. Our VIPs who attended here today, thank you. Our clinical department who will be out there, and importantly, that's going to be the next stage of our event, where you get to talk to more scientists and ask more questions. Also want to thank the education, with Greg Chin, who runs our education programs, is an important part of making this kind of event happen for the public, and our communications department run by Robin Snyder, who's sitting down here at the front. I'm going to close with a few comments. First of all, please stay connected with us. There's so many ways that you can do this through our website. We have our podcasts, we have online learning, we have our community seminar series, which I think possibly some of you or many of you have attended already. These are the kind of ways that we try and communicate what we do up here on the Hill to the local community and to the Bay Area and beyond. And we understand it's vital that we have to talk about what we're doing. And actually, the great thing about being at the Buck is you've heard 3 talks today that you could have heard at some other university, and they would have been amazing. But the one thing, the magic that happens here is that you've heard these talks in the one place, and you can talk about and ask about the connections between them. How does ketone bodies affect taking out the trash? What's going on about taking out the trash in space? Would ketone bodies actually benefit astronauts? So these things are being thought about all the time. The connections between different kinds of science are being thought about all the time at the Buck, and we do our best to try and communicate that. This event, let me come down here. This event was created by our trainees. This didn't come up from leadership or faculty or anything like that. Our trainees came up with this. And the faculty, the more senior scientists, are in absolute awe of the creativity and the energy and the optimism of these people and their colleagues. It's truly amazing. The career path right now for a scientist, well, it's always been difficult, but it's particularly difficult now. The career path is spending 70, 80 hours a week in lab or in front of a computer. And you're doing this at a time where you're looking after aging parents. You're perhaps building a family. You're trying to work out a way to buy a house. And your salary is quite miserable, to be honest. And so the challenges are extremely hard. And we, the scientific establishment, what we do is to give them a 6-foot bench in this building, and we ask them to do experiments that have never been done in the entire history of the world before. Not only that, we expect those experiments will be significant and innovative and change human health. And the miraculous thing is, they do it. They actually do it. So what do they need to help them? I mean, obviously, it's financial support, obviously. Your presence today is amazing, and it is so encouraging for them and for us that you come to an event like this. But please, when you leave this event, and we're all going to go in there. I want to make that point. We're going to go in there and look at posters and talk more science. But once you actually leave the building, please talk to your family and your friends and talk about these people, these trainees who have the ambition to change the world. They and their trainees will cure Alzheimer's, and they'll cure Parkinson's, and they'll cure cardiovascular disease and osteoporosis and all those diseases of late life. They will do it if they get a chance. Now, we've had serious conversations with trainees who are saying, well, I'm going to leave the USA, we're done here, or, I think I'll leave research. It's not going to happen. We're not going to allow it to happen. The buck is going to be here training people for the next 50 years. But we need your support and the support of your friends and family to do that. So that's it. Thank you so much for being here, and enjoy your time out there.